Thank you, Mark. So I'd like to talk about exogenous VOC probes, or what we call EVOC probes. So at Owlstone, we're obviously developing breath biopsy technologies, and we see tremendous application in early detection precision medicine. I think we're trying to do two things. One is use the technologies to develop tests ourselves, but also try and provide enabling technologies to the research community. So we do work a lot with academic and pharmaceutical partners. Let me just go over this. So two of the basic questions in VOCs and breath is, what are they? Where do they come from? So obviously some are of endogenous origins, produced in the body. So these endogenous metabolites have potential for use in different diagnostic applications. There's actually a lot of exogenous compounds there as well. So this is, pharmaceutical drugs, environmental exposure, dietary sources, those types of things. So I want to come back and talk more about the exogenous sources. The second question is, where do they come from? So obviously some are produced in the airways, but most originate from the blood, which makes sense. Your lungs exchange chemicals from blood to the airway. So one of the big advantages of breath is that you can pre-concentrate chemicals over time, so help drive sensitivity of tests. Breath has a lot of potential, and I think once we started looking at it, the kind of elephant in the room is, why is it not being used in clinic today if it's so great? It has obvious benefits for patients. And I think there's been, some historical challenges, things which are touched on this morning. How do you standardize some of the aspects of collection analysis? And most importantly, how do you build much larger studies, which are in representative clinical populations, where you are getting through to blinded validation? So I think there's a little bit of a lack of studies that have got to that point. But I do think there is obviously tremendous potential there, and some of the presentations this morning, again, are acknowledging we need to do more in terms of collection of bigger data sets. So, we've developed a series of technologies. I'm not going to do the sales pitch here. You can go and look at the kit out on the stalls, but it's a mix of collection and analysis. The one thing I would say is, because there are a lot of people use and receive antivirus products at the moment, we are developing next generation systems. So some of them are here, so clean air supplies, the kiosks, the kits, and those types of things. We would love your feedback. So, come and give it a proverbial kick of the tires. So I think, earlier this morning, we're talking a little bit about discovery type analysis. So one of the things that we do with our own studies is obviously try and find biomarkers related to different aspects of disease. But we do the same for partners. So pharmaceutical partners can get the hardware, send the samples, and we'll process those samples and try and find biomarkers. And this is, very conventional type workflows. Analyze GCMS, build a feature table, build a model, test that model. And I want to move through this quite quickly to get on to more of the targeted side of things. But one of the things that we're working very hard on is how we standardize absolutely every part of that, both in terms of the collection, the transport, what happens in the lab, and then the data pipelines that we use to spit models out the back and evaluate those models. So here, taking just a simple problem like smoker, non-smoker classification, where you get a feature table, looking to see what are the significant features, Volcano Plot, looking at the fold change between those different features. And then going through and looking within those features, doing tentative ID of those compounds, box plots for these so you can see the differences between case control, building this into a model, so using a lot of the tools that have been described already, random forests, making sure that you're doing things like cross-validation, and then ultimately doing much larger studies and blinded validation. That's the point at which I start to believe results. I tend to believe nothing until I see blinded validation. And then evaluation of those models, trying to get to the point of, what is the sensitivity, specificity of those types of models. So I think that is how, we do a lot of this. We do a lot of it with partners as well. And that is your kind of classic top-down, data-driven approach of look at everything, try and find what correlates to the different disease states. And we and others have published quite a lot using these types of approaches. I want to take a slightly different tack at the moment and start to talk a little bit more about targeted type analysis. So when you look into the breath field, what tests are deployed now, it is things like H. pylori testing. So typically where substrates have been administered and it ties back to a specific understandable bit of biology. So Anil mentioned, the use of labeled substrates, so sticking a C13 on the different compounds. that's metabolized and leads to the production of C13CO2 on breath. And also, as Anil alluded to, you can put a C13 onto different compounds or drugs, and it will be metabolized in specific ways. And this allows you to answer different questions. So Anil mentioned methionine, but you can look at things like CYP, P450 activity as well. So things like methathicin and those types of things. So, for me, the key is what's the biology? How do you probe that particular bit of biology? And there's been a lot of interesting papers published here looking at things like chronic liver disease, so different stages, cirrhosis, for instance. So where methathicin breath test does show a lot of utility here. So, to me, this is interesting. It points at a direction of travel here. So, one of the challenges, or a few of the challenges you get with the C13 strategies is you can only really look at one thing at a time. So you stick your C13 on the one compound and you're getting one readout. So you can't take cocktail-based approaches or answer multiple questions at the same time. Also, it's very expensive. And from a regulatory standpoint, it's very complex as well. So you need to get approval once you start to label some of these types of compounds. So going back to looking at, what are the VOCs on breath? So again, we have our endogenous sources, endpoint, metabolism, that's great. And then these exogenous sources here. And there's been a few interesting things that we've seen in the literature looking at dietary sources of compounds and how they're then metabolized. So here, trimethylamine, how this is metabolized in a different way from patients who have acute alcoholic hepatitis. So this is, it comes in via diet, there's a particular pathway there, and then you have this elevated concentration trimethylamine. Another paper which this really changed my view of what we can do on breath, and actually the people that did this work are in the room somewhere here, can't see them now. We had always considered exogenous compounds as noise, something to be eliminated in your analysis. And in this study, where limonene was coming in through dietary sources here, for patients that had liver damage, it wasn't being metabolized in the same way from perfectly healthy controls. Because limonene was lipophilic, what you get is essentially build up over time, and you would see significant differences between case control here for limonene, and then pre and post transplant, the limonene washes out again to normal levels. So this was interesting because it's pointing at how an exogenous compound could be used as a probe to ask a question about specific biology. So, the basic premise is rather than relying on it coming in by accident through dietary sources, administer a specific probe, which is volatile and therefore will be elevated on breath. But the key being you need to understand the biology that ties back to there. So, one of the things that we've been thinking about, working with Anil on this as well is, drugs are metabolized by a small handful of enzymes in the liver. We know people metabolize differently, and this can lead to toxicity and efficacy for different drugs. And there's pharmacogenomic tests today to tell you what type of metabolizer you are, but your phenotype changes from other drugs, lifestyle, other types of interactions as well. So, these drugs will be metabolized by things like CYP3A4, and, a lot of the work that the community's been doing, looking at peppermint washout, we were doing a lot of that work as well to try and establish technical variation of the system, how well the assay will perform particular compounds. But, these compounds are metabolized in exactly the same way as a drug. So you don't have any of it in your body to start with because it's by definition exogenous. If you ingest it through a peppermint capsule, you then get a rise to a peak and a washout profile there. And I think the key being that washout profile then relates back to a phenotype, so the activity of these particular liver enzymes. So we've been doing some initial experimentation. Again, it'd be great to get feedback from you all about this approach. So again, peppermint capsules, mix different terpenes in here. where you're looking at these compounds which are metabolized by different CYP enzymes, and then looking at some of the specific endogenous compounds. So you don't expect things like acetone and isoprene to change. You do expect, obviously, the exogenous compounds to change. What's really nice about this approach is when you look at the fold change, so again, because it shouldn't be there and you're introducing it to the system, A, you know what it is, but B, you can introduce it in very large concentration as well. So that is much easier than looking for small fold changes in endogenous metabolites, and actually where the endogenous metabolite might not give you the specificity that you need for a test anyway. So I think analytically being able to introduce a compound makes life easier from the point of view of running the assay. So we've done some follow-up work doing repeat sampling over a 5-week period with one individual. And then looking for specific compounds, so at baseline, so prior to ingestion of the capsule, peak, which moves around quite a lot actually. So one of the things that you need to figure out is, how do you, things like gastric clearance, if you've had food before, you need to think about when that peak is going to happen. So again, you don't expect to see differences with endogenous metabolites. You do expect to see differences with the exogenous probes here. And the basic principle, again, being the area under curve for the ratio of these things is starting to give you information about phenotype or organ function. And then, again, limonene. So you see here very significant change, obviously, between baseline to peak, and then the 3-hour plateau afterwards. So one of the other things that we did was looked at the limonene concentration in the breath of 136 people through our LUCID lung cancer study. And as you would expect, there's some background now through dietary sources, but with the exogenous probes, you can punch way above baseline by administering the compound there, which is represented by the orange circles here. So one experiment that we've done recently is starting to say, okay, well, if this is being metabolized by these types of enzymes, can you inhibit? So initial data using grapefruit juice as an inhibitor is suggesting that this may certainly be the case. This is early data. There's still a lot of work for us to do in terms of development of the assay, but ultimately we want to get to the point where we can use this as a research tool to start to answer these types of questions. But this is quite interesting because it shows that there is the potential here to, obviously, you're modifying that phenotype with the inhibitor. So I think here again, this points to the potential utility of using EVOC probes to answer specific questions. So again, breath's great because it's non-invasive, we all know that. The nice things about EVOC probes is you can start to think about cocktail approaches. And that's both in terms of the substrates, so the substrates that target different enzymes, but also the downstream metabolites as well. You can multiplex the assay, you can look for 10 things, 15 things, so you can pick and choose what bits of biology you want to probe there. They're super safe because they're basically food products, and also it's extremely cheap. So you get peppermint capsules for about £1 from Amazon. So I think there's some advantages that we see here to this type of approach. So you can start to think about phenotyping, but also organ function here as well. So NAFLD and NASH is particularly of interest because, again, you're relating back to organ function. The prevalence is astonishing, over 30% in a lot of countries. And the economic burden is also astonishing as well. So in the US, it's about $103 billion a year, and in the EU, it's about 35 billion euros a year. So we've been looking through literature, seeing some interesting results where people have been looking at things like microdosing of midazolam to evaluate CYP3A4 activity, and then relating that through to controls versus NASH again. So, there's a few things which are piecing together, which are interesting for us, and this is one of the, the aspects of the research we want to continue to push. And you can think about this sitting at different points within the clinical pathway as well. There's different places where a test like this may be useful, and it's obviously important to think about the health economic aspects of that and why there is actual clinical need, in addition to the development of the assays and those types of things. But our goal is to, again, work with the research community with these approaches here to help say, is there something worthwhile with these types of EVOC probe strategies? What it does do is change what we need to be thinking about a little bit more. Because, initially we're like, okay, you need the hardware, you need GCMS data analysis, you need to do machine learning. This is much more about you need to understand basic biology, you need to be identifying the target enzymes, design of those probes and substrates, and that's a very different set of skills. So we've been trying to build a team internally to help address some of these gaps that we had as well. But it does, fundamentally change the way in which you start to think about things. The big advantage, obviously, is that, when you're doing validation studies, you can run much smaller numbers because you're coming in with a targeted hypothesis as opposed to the standard fishing expedition, which by definition requires an enormous amount of samples because there's a lot of features that you're looking at there. So you fundamentally need to have a lot of samples. So this is also efficient from the point of view of running these clinical studies. And it leads across into other applications as well. So we're doing a lot of work in early cancer detection, running 3 big programs at the moment. LUCID, looking at lung, intercept colorectal cancer, and PAN, looking at 8 other tumor types. LUCID is the most advanced, so recruiting up to 4,000 patients, which again is the level of, that's the numbers you need to do robust discovery and validation. So we've recruited about 2,000 patients to date there. And that again is, top down, what are the features of interest, build a model, validate that model. But we now want to augment that with a more bottom-up approach of let's understand the biology, let's try and design evoke probes for this. And we're doing a lot of work with Christian on this as well. So, I'm an electronic engineer, so a lot of what Christian was talking about, I don't understand any of it. But I know you need to have that perspective on it to help, again, understand both how the endogenous metabolites might change, but also inform choice for substrates. So, part of what we are trying to do there is saying, let's try and narrow the search. And let's not start with, 1,000, 2,000 features. Let's be hypothesis-driven, look at endogenous metabolites, but you still have the fundamental challenge there that you probably don't have the specificity and the fold change are likely to be relatively small in those types of things. So it creates an analytical challenge. Whereas if you use the analogy of imaging, for instance, and me and Paul were just talking about this in the break, where if you think about imaging, PET scans, you administer a tracer today, and you stick someone in the scanner. There it is. This is the tumor's hungry for sugar, and you can see that once you're in the scanner. So can we use exogenous VOC probes here with a similar type of approach and saying by understanding the biology of the cancer, can we get a degree of specificity where we hit it with a substrate or probe, look at the decrease of that substrate and the downstream metabolites there. So there's enzymes which are of interest here, so things like aldoketoreductases, where again you're starting to think about both bottom-up but also the substrate design, where these things are not normally expressed in lungs. So as well as our discovery type models, we're thinking much more about substrate selection that allows us to target that particular type of biology. So this is work in progress, but it's a general direction of travel for us, where we're using this approach to augment what we're doing on the top-down side of things. So that's the end of the talk. Thank you to everyone at Owlstone for doing all the hard work, basically. They're the people that make it happen. But I would love to, again, get your feedback on this, answer any questions that you might have.
Thanks, Billy. I'm going to open the floor to some questions. Please introduce yourself.
Ivan Royd, Middlesex University. I haven't heard much about the possible application of technology to diagnosis of infectious disorders, in particular neglected tropical diseases. And if so, and in particularly if you can ever reduce the cost of the hardware in the same way they've done with mass spec, where you could get a 20K instrument that will do mass spec now. Any chance you can reduce the cost of the hardware so people in remote locations or GP point of care and so on would be possible?
I think the hardware and the technology bit is the easy bit, actually. So, what we were originally doing was developing sensor technologies for explosive and toxic gas detection. When we first started looking at breath, we're like, we want to develop tests for lung cancer, we'll go to literature, get reported VOC biomarkers, functionalize our famous device, brilliant, we're off to the races. The problem was, there's not very good biomarkers which have been validated for lung cancer there, which is why we had to go a step back and invest so heavily in a lot of the biomarker discovery capability that we have. So I see the breath field in two distinct phases. We're in phase 1, which is about can we identify and validate robust biomarkers? Because the reality is we don't have very many at the moment, certainly of the endogenous variety. And I think that's where we see that we need to be thinking, as a community, how do we solve that biomarker identification question? Once you have that, how you then implement, I think, is actually fairly straightforward, either in a central lab setting or in a near patient or even real-time point of care type setting. So those things exist. What they need is someone to come along and say, it's a panel of these 5 biomarkers that's been validated in a large study.
Just to comment on the exogenous probes, whether they're labeled, unlabeled, the most important thing is compliance with fasting.
Yeah.
If the patient doesn't comply with at least 8 hours of fasting, giving probes orally is just not feasible.
Yeah.
And basically 24-hour abstention of alcohol is also important because it inhibits the absorption of the substrate that is being given to the individual. Yeah,
no, we've certainly seen that as well. So the approach that we have at the moment is, pick the 3 compounds, try and reduce the pre-analytical analytical variability as much as possible. But in some of the initial washout experiments you're doing, you see that Cmax will move around the place as well. So I think that is a very important consideration. So I think once we pin down the assay, we're happy with that, we can then start to think about how do you address aspects like that. But totally agree with it. And the thing is, for the alcohol, for instance, that this is measuring the actual phenotype, which is of interest above and beyond what you can do just with the pharmacogenomic approaches.
It's on. Hi, I'm Rian. I'm a researcher working with cancer patients at the moment. So how do you see this logistically when, for example, diagnosing cancer, right? If you have a test that works, if you have a model that works, how do you see this logistically working in the clinic? Do you have any ideas about that? Yeah, so I think when we were first approaching it, let's assume you have some targeted compounds and you can functionalize it into our FAMES device or another platform. The conclusion that we've come to is that you don't need a real-time answer. So in early cancer detection, it matters you pick up the cancer at stage 1, not stage 4. But that doesn't matter if it's on a Monday or a Wednesday. So it is perfectly acceptable to collect a sample and send the sample for analysis, so long as the assay is good at picking up the early stage disease. So I think the perspective that we have is it's important to have a test which is acceptable to patients, low cost, but you can distribute that collection far and wide. So, beyond secondary care into primary care type settings and those types of things. So our view is that, with something like Receiver, for instance, you could put that anywhere, but the samples are coming back to be processed centrally because you can get economy of scale there in the test as opposed to having to distribute analyzers out into the wild. So I think that's how we see it. And it fits with what is done currently. If you look at colorectal cancer screening with FOBT or FIT, it's all lab-based. So I think but I'd love your perspective, I think for cancer detection, that's acceptable. In the case of infectious disease, that might not be acceptable because you obviously need a fast turnaround there. So I do see, if I may, I do see in the clinic that, for example, I'm not sure if this is applicable, right, but for head and neck patients, for example, when they experience symptoms and they get diagnosed, they need to start treatment within a matter of days, right? So I'm just wondering whether this is logistically possible to send it to you guys and then get a result back. You know what I mean? Yes, yeah. The short answer is yes, but I think the problem is contained in what you said there. Once they get symptoms, they need to go on the treatment faster. So if you think about lung cancer, for instance, you need to be in the at-risk population where you're asymptomatic. That's where you'll make the decisive difference to the patient outcome. So I think there it's using the right risk criteria
and being able to collect sample or run a test, which isn't necessarily a definitive diagnosis, but it's funnily down there to say, okay, you might do a breath test that is suggestive of there being something there, and you'll still need to go for gold standard workup, CT scan, and the like. But I think the key is asymptomatic populations.
Time for one brief last question.
I'm Gudrun Koppen from Belgium, from the Flemish Institute of Technological Research. I had a question on if you sample from patients, do you systematically use different sampling conditions, like not eating 2 hours beforehand, sitting upright, being quiet, something like that? Do you have some sort of standard protocol for this?
So in the LUCID study, we don't. So the LUCID study, we get patients when they're referred under clinical suspicion. So there might be an incidental finding, or they come through 2-week wait and they're going to Papworth or wherever. So, that's trying to be reflective of how you would see the patient if the tests were actually deployed as well. So we don't apply a lot of those constraints. That would be different in phenotyping or organ function type test, where we think actually you do need to do things like fasting overnight or whatever may be the case. But the challenge is, if you become, obviously the breath varies a lot. It is reflective of phenotype, and that's part of the power of the technique, but part of the challenge as well. But if you apply too many constraints to the patient before you can collect a good sample, then it negates a lot of the advantage of the approach, I would say. But it depends a lot on the clinical question you're trying to answer.
Thank you, Billy.